Okay, great. I think we will get started. So I want to welcome everyone to our webinar tonight, Water Quality in the Merrimack, a 2021 review. We're all very excited to be here. This is a bit of a culmination of many years, but at least 1 year's worth of work collecting water quality data in the Merrimack, using great time from our volunteers and efforts from our staff. And we've been using that time to pull together a report and now this webinar and some new dashboards that we'll be showcasing tonight. So we're very excited to be here and want to thank you all, panelists and participants, for taking time in your evening to share it with us. So my name is Lauren Zielinski. I am the New Hampshire Water Resources Program Manager with the Merrimack River Watershed Council, and I'll be moderating tonight's panel. And I'm also joined by Julia Halm, who is our Community Engagement Coordinator, and she'll be helping me monitor the chat and the Q&A box. Just a quick reminder from the start, this will be showcasing our results from this year, and then we'll have our experts on the panel, who I'll introduce shortly, talk about some different topics. But if you have questions of your own, please put them in the Q&A box, and we'll make sure that we get them to our panel and we can discuss them as a group. So like I said, this is our effort to showcase the work that's been going on this year. As part of this work, we have 13 monitoring sites in Massachusetts, and also added a couple sites in Manchester, New Hampshire this year, and have been collecting different data on physical and chemical parameters, as well as bacteria monitoring. And we have some interesting results to share. So we're happy to be here with you all and to let you know what we found out. And so first, I would like to introduce our panel. We have different colleagues of ours and partners that we work with closely, and we thought this was a really nice, diverse group to get together and discuss different topics around water quality and the Merrimack and how things are going and what the future is looking like. So first, I would like to introduce Jill Carr from Mass Bay's National Estuary Partnership, and she can tell a little bit about herself.
Hi, good evening, everyone. Thanks so much for having me, Lauren and colleagues. My name is Jill. I am the Coastal Data Scientist at the Mass Bay's National Estuary Partnership. And what I do there is specialize in developing monitoring plans, both for water quality, habitat mapping, and other things like that throughout the estuaries in our embayment, and also go up into the rivers as well. So I have a particular interest in the Merrimack River, being a resident of the Merrimack Valley in Groveland, Massachusetts. So I've really enjoyed following along with MRWC's work, and I really look forward to this presentation.
Great, thanks, Jill. It's a pleasure to have you here tonight with us. Next, I'd like to introduce Chris August. He works for ACV Environmental and is also an MRWC board member.
Hello, everyone. Chris August. I'm currently working for ACV Environmental as an environmental manager for one of their treatment and hazardous waste chemical treatment plants out of New Jersey. I've experienced working in the greater Boston area, working with industrial pretreatment users in regards to wastewater. And the Merrimack River is definitely something that I care about because I used to live in Haverhill, Massachusetts. And I enjoyed helping out with the board any way possible that I can, even though I am working from a distance.
Thanks, Chris. We know life takes us in different locations and directions, so we're happy to have you with us, even though you've physically moved out of the Merrimack. And next, I'd like to introduce Wendy Hager-Bernays, and she is with the BU School of Public Health.
Hi, I'm Wendy Hager-Bernays. I am a clinical professor of environmental health, and we have been partnered with MRWC now for several years. What we do is we're really interested and we study the impacts of the river on health, on people's health. So we have been working with MRWC in working with the data, doing some collections ourselves, and along with doctoral candidate Beth Haley. This has been a very, very productive partnership. I've said that 3 times. I'm sorry. I'm so excited to be here that I'll just stop at that. So thank you very much.
Thanks, Wendy. Outside of your personal enthusiasm, it's also great to kind of showcase working with academic partners as well as state and private partners. So I think it's very cool to have a nice mix of people here on our panel tonight. And next, I'd like to introduce John McCone. He works with us at the Merrimack River Watershed Council as our policy and education specialist.
Hi, everyone. Thanks for coming tonight. My name's John McCone. I've been with MRWC for about 4 years, and I work on education programs. I do a lot of presentations, gather a lot of info, work with some of our local partners, state and federal and local as well. And just really love what we do. You know, this is our river. We all own it. And we're the people who are trying to protect it. Everyone who's here on this panel and folks who are participants here and so many other people in the Merrimack Valley. So thank you for everyone who supports us. We really appreciate that.
Yeah, thanks, John. And I like your point. This is our home. This is where we want to be and use the river. And we all play special roles, big or small, to help protect it and make it a better place. And last but not least, I'd like to introduce Susie Bresney. She is our Massachusetts Water Resources Program Manager here at the Merrimack River Watershed Council.
Hi, everyone. Thanks again for coming tonight and for listening to this presentation and this panel. A super important topic at the Watershed Council. I oversee this water quality monitoring program, as well as work with different partners, municipalities, other NGOs, state and federal agencies to improve water quality in the Merrimack through a variety of ways. So this is a topic that's really important to me and important to our organization. So I'm really glad to see you all came out to listen. So thanks again.
Thanks, Susie. I think many of you know Susie and our other colleague, Jose Tapia, and they do a lot of the hands-on water quality monitoring throughout the year. So I think we'd like to give them a big thanks for all their hard work and effort, and in addition to all of our volunteers that go out and collect our samples for us. And with that, so that's our panel. First, we're going to have Susie give us a quick overview of our results from our 2021 monitoring season. This is a recap of the report that we published on our website, which is in more detail. So if you have questions, we encourage you to go take a look at the report, because we can't cover everything in 20 minutes, but Susie's going to do her best to give us a great overview. And if you have any questions, please put them in the Q&A box, and we will be sure to include them in our panel after Susie is done.
Great. Thank you so much, Lauren. Let me see if I can get this set up here. Can you see my screen?
Yes, looks good.
Okay, great. Wonderful. So like Lauren said, I'm going to do a quick overview of the data and analysis that we've done, the data that we've collected in 2021 on water quality in the Merrimack. I'll go over a bit about our monitoring program and some background information, talk a bit about the environmental conditions of 2021, which provides really important context within which we can analyze and understand the data that we've collected. I'll give you a quick tutorial of the dashboard that Lauren's developed and is available for you to explore all of the data that we've collected. This will be just a brief overview, but you can dig in yourselves if you'd like. I'll also show you where to find the report if you haven't read it already. We'll go over some watershed-wide results, and then we'll talk about some data-driven solutions that we're working on here at MRWC. So a quick overview of our program. We monitor for 2 different types of information. We collect data on in-situ river conditions for ecological health, and we also collect samples for bacteria analysis to monitor bacteria concentrations for human health. And the in-situ monitoring, what we collect, right at the time that the volunteers arrive at the river while standing right there in the river, we collect them with these meters that you can see on the right-hand side here. We collect data pH, specific conductivity, salinity, total dissolved solids, and temperature. We're not going to talk a whole lot about these tonight, actually, for a good reason. Because all of the information we've collected demonstrated that these parameters are within safe limits for supporting good ecological health within the river. So a good result, but not a lot to dig into. What we're really going to focus on tonight and talk about, at least in this presentation, are the samples that we collect for laboratory analysis. We look at 2 types of fecal indicator bacteria, E. coli and Enterococcus. And these 2 types of bacteria tell us if the water has been contaminated with some type of fecal matter. So what does all of that mean? What are these bacteria? Where do they come from? Why do we care? How do they get in the river? There's generally two kind of types of bacteria sources for how bacteria get into the Merrimack: point source pollution and non-point source pollution. So in the category of point source pollution, hence the name, it's a source that you can point to. It's a pipe. You know, it's in the Merrimack, it's combined sewer overflows. So when a combination of stormwater and sewage enters our stormwater system and our wastewater treatment system, and it gets overflowed, it backs up into the Merrimack. So combined sewer overflows are one point source. Illicit sewer connections or leaking pipes or poorly maintained septic systems are also other point sources. Oftentimes these point sources present a greater risk to human health, not because they're point sources, but because of the bacteria that the water that comes from these sources contains is usually sourced from human fecal matter, and that presents a greater risk to human health. These can be mitigated by fixing the failing infrastructure, fixing the leaking pipe, changing the wastewater treatment system to mitigate combined sewer overflows, or completely diverting the stormwater so that it doesn't enter the stormwater system or the combined sewer system in the first place, to remove the potential for a combined sewer overflow to happen. So these are really infrastructural focused solutions. Whereas non-point source pollution is more bacteria that comes from a variety of locations. There's no single location that you can point to. So this could be bacteria from feces of animals like wildlife or our pets if we don't pick up after them. Also manure that's spread on agricultural land for fertilizer can have bacteria in it that makes its way into the river. And these are really driven by rain events. So when it rains, that's what carries the bacteria from these various sources into the river. These often present a lower risk to human health because the bacteria is sourced from animals as opposed to human fecal matter. And they can often be mitigated with things like green infrastructure. So using nature to filter out the water before it reaches the river, rather than sending that water directly into the river. So we try to look at these bacteria sources and understand them with our water monitoring program in two different ways. So in space, we try to look at trends in bacteria across the 13 monitoring locations where we sample regularly. So you can see on this map has our 13 locations. The little yellow triangles are combined sewer overflow outfalls. So that's where combined sewer overflows contribute to the river. So we've located our sites up and downstream of those. This year we added 3 new sites, 2 in Manchester and 1 in Lowell. Important to note because we started sampling there in July. So we have a smaller data set for these sites than we do all of the other sites. And we moved 1 site in Drake a little bit downstream to improve access for our volunteers. We also try to understand these bacteria sources by sampling in time. So we sample during our regular sampling program once a month in the winter and twice monthly the rest of the year. These are days that are set at the beginning of the year and they're not influenced by weather. We sample no matter the weather. And then we also sample following CSO events. In 2021, we sampled after 3 large combined sewer overflow events. And we sampled 4 to 5 days continuously following that event to see how bacteria moved through the system. We have 36 amazing volunteers who collect our samples, some who visit sites regularly for a regular sampling program, and some who show up with very little notice and go out there and collect samples after CSO events, which literally this program absolutely could not happen. We would not have any of this data without our volunteers. So we're so grateful to them. In total, we collected 312 samples that were analyzed in 2021. We analyzed these samples for E. coli across all of our sites and for Enterococcus at the brackish sites. And that's because it's a better indicator for sites that have a higher salt content in the water as opposed to E. coli. We did start monitoring for Enterococcus at all of our sites in July, but we won't dig into that data again since it's a small data set so far. So to talk a little bit about the environmental conditions of 2021 and put a little context into the data that we're collecting and allows us to kind of understand how we can analyze it and what conclusions we can pull from it. So this graph shows some historic rainfall data from the gauge that's in Lawrence. The dotted line shows the historical monthly maximum for rainfall, and then the dashed line shows the historic minimum. And the two lines in between are different averages. So a good point of reference is 2006, which was the year of the Mother's Day flood, which I think a lot of people have in their memories. You can see that the 2006 and when the Mother's Day flood happened set the record for the highest total rainfall in May. So it reached that historical maximum line. And here we are in 2021, where we see that the July rainfall exceeded that historical maximum since 1953 and was almost as much rain as we saw during the Mother's Day flood. And in the months following, we saw well above average rainfall in those months. So really important context to consider because rain is definitely a driving factor for a lot of the bacteria contributions to the river, either through CSOs or non-point source pollution. As a result, we saw a record high for monthly CSO volume that contributed to the river. So this is a short record just since 2018, but still we see 2021 saw really high total volume of combined sewer overflows flowing into the river. That was a result of that record high rainfall that we saw in July. We also see some of the higher contributions of combined sewer overflows in the months following July as well, linked to that above average rainfall. In terms of annual contributions of combined sewer overflows to the river, we saw the greatest in 2021 since our record of 2013 that we're working with, 822 million gallons of combined sewer overflows in total were contributed into the Merrimack, or the main stem of the Merrimack, in 2021. Another important piece of context that I want to mention is that Lowell and Manchester are the two largest contributors in terms of volume to the river. So they each contribute about 41% of the total combined sewer overflow volume to the river each year. Haverhill contributes a small amount in terms of volume, but contributes the greatest number of individual events. So there are frequent but small events in Haverhill as compared to other locations. I'll give you a quick tutorial of the dashboard. Let me just pull up another screen here. And this will help to orient you to the different types of graphs that you'll see throughout this presentation, and let you know where you can find more information, since, like I said, we won't be able to go through all of the details of all of the samples that we've collected today. If somebody could just let me know, Lauren or Julia, can you see my the website?
Yes, I can see the website.
Perfect, thanks. Okay, so you can find the dashboard here on our website, merrimack.org, and you click on Programs, Science, Water Quality Monitoring Program. And that will bring you to this water quality monitoring page where you can see some information about our program. And you can see these dashboards that we've developed. So this is an interactive way to explore all the data that we've collected in the Merrimack in 2021. So you see we have two dashboards. This is one with bacteria data, and this is one with that those other parameters linked to ecological health. So if we take a look at this bacteria dashboard, you see a list of all of our sites here on the left, a map of those sites here in the middle, a photo of the site on the bottom. And in the center section is a summary of all of the E. coli and Enterococcus samples that we've collected at those sites. Light blue shows samples that were within the limit that the EPA has set for safe recreational use. and red is above that limit for safe recreational use. So that's an indicator to tell us kind of what the conditions of the river are. And then over on the right, we have 2 time series graphs, which show every individual sample that we've collected. The blue ones were collected during wet weather and the yellow were collected during dry weather. And these gray markers show just where a CSO event occurred upstream of that site. So you can click on different sites and the graphs will change, showing you the data from that site. And then you can hover over the different samples to see exactly the concentration of bacteria that were collected. You can click to just view wet weather samples or just view dry weather samples. And you can see here a summary of all the samples collected from that site, how many were above the safe limit in red in both wet weather and in dry weather, and how many were within the safe limit in light blue in both wet weather and dry weather. Our second dashboard here is this one shows the different parameters that we measure in situ or right in the river with handheld meters. We measure pH, conductivity, salinity, total dissolved solids, and temperature. And again, you can explore these by site. So you can just click on one site and view the different graphs. These blue areas indicate where the levels support freshwater fisheries or freshwater aquatic organisms. So you can see if the values at each of the sites are within those limits. So that's a quick overview of the dashboard. Definitely encourage you to come to our website and explore more, check things out. Let us know if you have questions. And then also encourage you, if you haven't already, to download our full report, which is here. You can download the full report and review more detailed information than we'll have time to present here. Okay, now let me just go ahead and I've got to share back to the presentation. And let me make this full screen. Okay, are you able to see my screen again, the presentation?
Yes, looks good.
Great. Okay, so here's an overview, and this is one of those graphs that you saw there on the dashboard that I showed. Here's an overview of the results across all of the samples that we collected, and specifically all of the samples that we collected during our regular monitoring program. So this is the program, the dates that were previously decided. They're not dates that we selected when a CSO occurred and we said, okay, we got to get out there and sample due to a CSO. So we see some really promising stuff. The very first thing that we see is that the vast majority of the samples, 80%, are within safe levels. That's really exciting for us because we feel that the Merrimack is a place that everyone should enjoy and should go out and recreate on. And these results kind of reconfirm that. What we also see is there are a fair number of unsafe samples, and some of those are during dry weather conditions and some are during wet weather conditions. So this tells us that there are likely multiple sources of bacteria contributing to the river, some non-point source and combined sewer overflows driven by wet weather, but also probably some illicit sewer connections or other sources that are not driven by weather. If we look at these results in space, we see on the left-hand side here is the Manchester site, the most upstream site. All the way to the right-hand side is the most downstream site at Salisbury Beach State Reservation. We see that the distribution of light blue and red, or safe and unsafe samples, is somewhat even across the site. So most of the sites have a mix of a few unsafe samples and mostly safe samples. However, the Lawrence Boathouse site had no unsafe samples during our regular sampling. So that's an interesting finding. Additionally, the Methuen-Riverview Boulevard site had the greatest number or the greatest ratio of unsafe to safe samples. And that's consistent with our findings last year that we suspect there's an illicit sewer connection upstream of this site. And we have confirmed that with the EPA that they have found a source and they are working on resolving that. So that's good news. And that's why we do what we're doing is to find solutions to some of these issues. Yes, and now if we take a look at the results in time, so across the months, over on the left-hand side is January, to the right is December. At the top, we have E. coli for all of our freshwater sites, and at the bottom, we have enterococcus results for our brackish water sites. So what we see is there is the majority of the samples that were within the above the safe limit, or the unsafe samples, occur from July to October. If you remember back to the graph we previously saw, that's also when we had the most rainfall and we had record or above average rainfall. So this says to me that these things are likely pretty linked, that a lot of these bacteria sources are driven by rainfall, either from CSO events or non-point source pollution and runoff. We also sampled after 3 combined sewer overflow events. So one in April, one in July, and one in October. And this table here shows where each of the CSOs occurred on each of those dates, how big they were, and then the number of days that we monitored following that event. I do want to note that we don't have Manchester listed on this table. It's not because there was no CSO in Manchester, it's because Manchester doesn't share data on individual CSO events. So we presume that there was a CSO at Manchester at each of these dates. However, we don't have the data currently to confirm that. So I just wanted to make sure I point that out. And so what we found is what we expected. There's a greater percentage of not safe samples relative to safe samples in within 4 to 5 days following a CSO event, both for E. coli and for enterococcus. And when we look at these in space, or from upstream on the left to downstream on the right, what we see across all of the freshwater sites is that all of the sites seem to be equally impacted by the CSO events, or there seems to be a similar ratio of not safe to safe samples at each site. No one site is completely red or completely blue. Alternatively, when we look at our downstream sites, the brackish sites, so from Amesbury down to Salisbury Beach and Plum Island, we see a different trend. We see that there's a lot more unsafe samples from the Amesbury site, and then as you move downstream, there are fewer and fewer unsafe samples at each site. So for the upstream sites, if you can remember from the map, there are many CSO outfalls mixed in among the sites. So as you move from upstream to downstream, there's not just one source of bacteria from CSOs, but there's continuous sources as you move downstream. However, these brackish water sites are downstream of all of the CSO outfalls. So what we're seeing here likely is that all of the bacteria gets contributed upstream of these sites, and then the bacteria dies off, or it settles out to the bottom, or different processes result in the bacteria concentration decreasing as you move downstream. Tidal influence could be a part of that as well. So based on these data, it looks like our Salisbury Beach site is least impacted by CSOs compared to the rest of our sites. So that was a really quick overview of our findings, kind of watershed wide. I want to leave plenty of time for discussion and all of your questions, which I'm happy to answer. But a few solutions to not leave you just with the leave you just with the sad stuff, but a few solutions that we're working on to resolve some of the bacteria in the river are that we'd like to better understand the transport and fate of bacteria from CSOs. So not just if a CSO happened upstream, but really, comparing the large CSOs with the small CSOs and how they might have different impacts. There are so many factors that influence how bacteria moves downstream. So we really want to better understand that. And some ways we're doing that is, more frequent CSO monitoring would definitely give us more data. This is still a pretty small data set. We only sampled after 3 CSO events. So more frequent CSO monitoring could contribute more data to help us unravel some of these things. We're also working with Wendy and Beth at the BU School of Public Health to, they're developing an E. coli model to estimate bacteria conditions based on rain patterns, CSO locations, and other data to better correlate different environmental conditions with bacteria concentrations. And we're working with other watershed organizations to share data on the main stem of the Merrimack River. So not just using our one data set, but combining multiple data sets so that we can have a bigger picture of what's going on. We're working on developing watershed-based plans in the sub-watersheds of the Merrimack, and these will be used to implement green infrastructure. So that will reduce stormwater runoff and treat stormwater with nature solutions within the soil, but it will also help divert stormwater so that it doesn't go into the stormwater systems and contribute to the existing issue of combined sewer overflows, particularly as climate change brings larger storm events and more rain. And so we're also working on, oh goodness, it looks like my last point here got a little muddled, but I'll just tell you what it says. We're working with state and federal representatives to advocate for federal funding to be channeled to the Merrimack Valley and specifically to solving CSO issues in the Merrimack. So we know how hard our wastewater treatment plants work to keep the Merrimack clean and to treat all of the water that's sent their way. But the problem is just so big that it requires federal funding to really solve. So we're working on making sure that they have the funding available to really achieve the solutions that they need. So with that, I'll say thank you. I know that was a lot of information quickly, but I'm looking forward to hearing your questions and diving in more with the panel.
Great. Thanks so much, Susie. We really appreciate it. Like I said, it's so hard to put in 1 year's of work into a 20-minute presentation. So we appreciate all your efforts doing that. We have some great questions in the Q&A so far. We also have some other questions that have been coming our way throughout the past year from volunteers, from people who live in the Merrimack, even some discussions that we've had as staff members as we've done our analyses. So we're going to start with a couple of these and then just kind of have a nice discussion with our panel members. So I'll invite our panel to come back and turn their cameras on, and I'll start shooting some questions your way. I think a good place to start is this term bacteria. We do a lot of monitoring around bacteria. In our report, we note that bacteria is actually short for fecal indicator bacteria, which is a little bit more of a complicated topic. So I'd like to send this question over to Wendy and kind of describe what is fecal indicator bacteria and what does that mean for risk to human health or human illness?
Sure, thanks a lot. First of all, that was an amazing presentation, Susie. Thank you so much. It was great to hear it all put together. So fecal indicator bacteria, or FIBs, are the organisms that we measure, like you've measured, we measure, to determine whether the water may contain feces. So the FIBs are indicating the presence of feces, but not whose feces. It could be dog, it could be goose, could be human. The FIBs, and the other thing about the FIBs, they're indicators, right? So we want indicators that tell us that there's potentially fecal matter there. But these FIBs themselves don't cause disease, right? They are not pathogenic, and they're relatively easy to grow in the laboratory. And so for those two reasons, that's why they're used, right? We can grow them. We can use them as indicating the presence of feces, but we don't know whose feces. So shall I keep going?
Sure.
Or shall I say? Okay, all right. So there are methods available that can better determine whether those species are human or animal. And these are called microbial source trackers. And Beth we will be looking at a human-specific source tracker, which is actually called HF183, which is the most commonly used sewage-associated marker, because these organisms are highly abundant and have relatively high sensitivity and specificity to human feces. So, and a little factoid, let me tell you something, one other thing, then I'll stop. So there's this really cool virus, it's called PMMOV, or pepper mottle virus, is the most abundant virus identified in the feces of healthy people. So this virus, right, is so cool. It's actually a plant virus, right? And it infects pepper plants. And when it's in human feces, because we eat a lot of peppers and pepper-derived foods, and this virus is fairly reliable to track the human feces. It's rarely, if ever, found in animal feces. And I'll stop there.
Okay. So no, I mean, it goes to show it's a challenge that we've had trying to figure out the source, like Susie was talking about, different sources geographically in different parts of the watershed, but also who is making that source or what is making that source is also a big question. So we're very excited to be working with BU and try and pick apart the details of those challenging questions.
Thank you.
Awesome. Well, thanks so much. Kind of in line with that, if you remember Susie's map, she had little yellow exclamation points kind of all throughout the river, especially around the urban areas in New Hampshire and Massachusetts. I'm going to give this question to John. Is there a particular region or particular CSO outfall that is of greatest concern or that has the most impact on the river, or is there kind of an equal distribution of impact?
Sure. Okay. So one of the slides Susie put up showed the percentage of where this CSO volume is coming from. So it's almost equally split right now between Manchester and Lowell. About 80% of the entire volume is coming from those two communities. And within those communities, there's two spots in particular. Might be good for fishermen to know about. One of them is right in Manchester, right behind the Fisher Cat Stadium. That's where 90% of it is coming from. And if you're in Lowell, right down around near where the sewage treatment plant is, Duck Island, there's a major source right there as well. So if you're in the river and you're maybe upstream from there, you're in better shape. Some of these other locations we talked about, like in Lawrence in particular, there's one actually very close to our office at the confluence of the Spigot and Merrimack Rivers. That's the heaviest source. And in Haverhill, it's actually underground. It's right under the center of the city where the little river goes underground into a pipe. So you would never really be able to see it or come in direct contact with it.
Great, thanks. Yeah, it goes to show the importance of knowing your watershed and finding out this information. I would say as a general member of the public, I might not know this, but it's great to have someone like John on our team who's able to let us know these things. John, you did mention about wastewater treatment plants. So I would like to send Chris a question. We've been talking a lot about CSOs, which typically is untreated water that goes into the river, but most of the water during normal time goes into a wastewater treatment plant and then gets returned to the river. So Chris, could you talk a little bit about how these wastewater treatment plants work and what is the quality of water that's being released, let's say in general, not at specific plants, but being released after processing from these wastewater treatment plants?
Yes, definitely. So how wastewater treatment plants work exactly are that they receive different, they receive water from different sources, industrial sources, rainwater, runoff, domestic sources as well too. All this kind of gets sent to the wastewater treatment plants. From there, this wastewater is treated through the different methods. So first off, you have primary treatment. And primary treatment, essentially what they do is it removes any solids, things of that nature from the wastewater to different processes. And the secondary treatment involves removing different biological hazards from wastewater effluent as well too. And the final stage of this involves adding chlorine or another UV light to kind of get rid of any other biological or chemical matter that may be in wastewater as well too. So that this effluent that is released back into our rivers is fairly clean for the most part. And this effluent is clean, and it's checked through different standards as well too. So wastewater treatment plants are required to get a permit, and through this permit, they have strict guidelines that they are supposed to follow in regards to monitoring, sampling, and as well as reporting these results as well too. And these wastewater treatment plants check for different things such as pH, the temperature, your limits, your nutrients in the water, your biology, your COD, BOD, biological hazards, as well as bacteria in the water as well too. And they check this to ensure that the water is essentially safe, that it enters our river. And this is very crucial because if there are any fluctuations or high levels of different hazards that go into our waters, this will affect the wildlife and also the water for this, how we wouldn't be able to safely use it for recreational use as well too. So it's crucial that this water is being treated effectively by our wastewater treatment plants.
Great. Yeah. I mean, it shows that, you know, we're working well and our wastewater treatment plants are operating properly. We're really, you know, maintaining the good quality of the river water while, you know, cleaning kind of all of the things, the human-made things that come inside of it. And it shows the importance of preventing CSOs, right? So if we can reduce the amount of runoff or the timing of the runoff and slow it down so more of that water can go into the treatment plant, the better off we are for water quality.
Exactly. The more we can do ourselves on our end to kind of reduce the amount of runoff or anything else that goes to a wastewater treatment plant, the better. I know for industrial users, for example, which I dealt with when I'm in the Boston area, industrial users often have their own permits that they have to adhere by as well too before they send their effluent to a wastewater treatment plant as well. And they kind of outline the same things to look for, such as, you know, the nutrients, your pH, temperature, BOD, COD as well too, to ensure that these levels are low enough so that when they do go to a wastewater treatment plant, they don't have to, it doesn't negatively affect the treatment process. And also so the wastewater treatment plant doesn't have to work as hard to remove these items from the water that's being discharged to our rivers.
Right. Yeah, and we're partners in this effort. So I'd also like to point out that we partner with different wastewater treatment plants and sanitary districts along the Merrimack to share data and discuss ideas and things like that. So we're all on the same team, moving in the same direction to get a clean Merrimack. A little bit of a similar note, this is actually from one of our Q&A questions. I'm going to direct this towards Jill. Someone was interested in using oysters to use as a, you know, filtering the river water. I know this is something that's happened in other estuaries. I know Great Bay in New Hampshire has a program through the Nature Conservancy. Jill, what are your thoughts on this and kind of the pros and cons of using oysters to help filter river water?
Sure. So prior to working at Mass Bays, I worked at Mass DMF, the Division of Marine Fisheries, for a long time. And we had lots of questions like this about using shellfish to clean the water, especially in areas that were prohibited for shellfishing. And they're prohibited because the water is impaired or unsafe. And in terms of being able to get permits, I think it might be insurmountable. It's a policy of theirs that no one will grow shellfish in a closed, prohibited area for public safety reasons, because once you start growing them, especially oysters, which are very costly and very delicious, folks come out and want to consume what you're growing or consume anything that might escape from your cages. So they have a lot of reasons why they would not allow that in the Merrimack at this time. But say it were to not be prohibited any longer, you'd still have challenges there too. From what I understand, you'd pretty much need to blanket the entire riverbed at the estuary end of the river in order to actually have a lasting and real effect on water quality in the river. It does work in some places better than others, really enclosed embayments. But at the estuary end, conditions are a little bit better there, perhaps, just because it's already more flushed. You have tidal exchange every day that's sort of bringing in cleaner water. So the bang for your buck there with the project in the Merrimack, while it is exciting, and I think it would drum up a lot of public interest and support, I think it would be pretty tough. I think you'd have a lot of challenges and might not get the results you're hoping for.
Yeah, sometimes those real world situations draw some challenges for you, but I love the creativity. I think it's really great using these natural systems to clean up our water. So I think this is a great idea, and let's keep being creative on how we can improve the water quality in the Merrimack. There's another question in the Q&A, and I'm going to direct this towards Susie about CSOs in winter. So we've kind of focused on CSOs in summer and rain events, but what's going on in the winter? Are they also of risk or of concern compared to those that we've discussed in the summer times?
Yeah, great question, and very relevant for the winter that we've been having. So I think historically the winter hasn't been the time period where the majority of the CSOs happen, because typically our precipitation in winter comes in snow, and snow doesn't create the runoff immediately the way rain does. So it's stored as snow on the surface and then slowly melts over time, but can lead to CSOs in the spring if all of that snow does melt at once. However, this year is a good example of kind of the winters that we might expect to see more often, which have more rain compared to snow, especially down in the Massachusetts area, and it might have snow and then melt and then snow and then melt differently than in the past when we had a lot colder winters. So we don't monitor for CSOs in the winter because our program is really focused on kind of understanding risks to human health, and there's not a lot of recreational use of the river in the winter. There's certainly some, and I know people fish year round, but not as much as in the summer and in fall and spring. But that's not to say that with times changing, with the climate changing, that this won't become a bigger issue and something that we want to think about for the future. We've definitely seen more CSOs this winter from the different weather patterns that we've been having. So great question.
Yeah, and very relevant to our changing climate patterns in this region, something that we've been talking about a lot internally as well. Another question from the Q&A. You guys are having great questions here. Paulina said she loves the dashboard. Really cool that we can see kind of data from places where we live or different regions and stuff. But curious, is this related to a CSO notification program? And is there a way that either the monitoring program in general or the dashboard is going to improve our knowledge of or timing of sending out these notifications? I think there's a lot of work happening in this space. So I think I'm going to send it to Wendy first to talk about the model that her and Beth are working on, and then maybe to Susie or John to discuss what we're already doing for notifications and how this model or some of this new information will help our notification program in the future.
So I will address the question of the modeling, right? Because the modeling will then be able to make some better predictions, I think, about what's happening downstream. That's the intention of the model. And that model will take into account the multiple factors that Susie both spoke about and alluded to, which have to do with volumes and temperature and rate of flow of the river and dilution, and all of these factors that are important in making predictions about when when and how I'll call it the bacteria move down the river. And so right now we're looking at these snapshots of what's happening, but we know that all these factors are involved in where the levels are going to go up and down, and what the time frame is over which that occurs. So the model will You know, it's not a panacea. It's not going to solve all of our climate and, you know, CSO problems, but we're hopeful that it will be very useful in making some of these predictions, and so that warnings and notifications can be more timely as a result of these predictions.
Yeah, thanks, Wendy. I know in the modeling world, there's a phrase that goes, all models are wrong, but some are helpful.
Yes.
So we're looking for a helpful model. John, do you want to discuss, you know, what MRWC is currently doing for our notification program, and how maybe this model or future insight into bacterial conditions will change our systems?
Sure. Okay. So I'll start out by saying that, you know, we talked a lot of other watershed groups, and we see what goes on other rivers. And I have to say that our water treatment plants, sewage treatment plants on the Merrimack have been much more responsive than what I hear in other watersheds. So we're really grateful to that. They, you know, we have a good relationship with them, and they understand what we're trying to do. And, you know, they're not certainly not the bad guys in any of this. You know, they're doing a great job with what they do, but they've got this old infrastructure that they're dealing with, and they need money to fix these problems. And that's one of the things we're trying to do, is bring them the money, help to direct it to them. So in terms of notification, the plants send us emails to let us know. They send us a fair amount of information. We'd like to see a little bit more coming from them, particularly when it comes to volume. You know, we'd like to know what the volume is as soon as possible, so that we know if it's a really bad CSO event. Like, Susie had a slide that showed Lowell had an 83 million gallon event. That's huge. And some of these events are very small, like 2,000 or 3,000 gallons. So we like to be able to get that gauge so that we know just how bad it is, so that we can inform the public. You know, this is going to be a serious event. You really want to be out of the river. One of the things we're doing now is we're trying to get a bill passed through the Massachusetts legislature that would have the DEP do studies, water sampling after CSO events at different places along the river, hopefully places where people are using the river, like boat ramps and beaches, things like that, and getting a sense to augment the data that we're collecting of how CSO, you know, contaminants are moving down the river, and then build these predictive models, and then use those to give people a real sense of what the river's like after a CSO, you know, because I think some of this data shows that maybe it takes a few days for a place to be to hit some really high levels, and the general guidance is 48 hours after an event. So that's not really a great measure all the time. We also see CSOs where we don't really see that much of an impact. So maybe those are times when people can feel safe about being on the river.
Great, thanks. I know many people either kayak, some people swim, or they fish, and they're very concerned, you know, what's going to happen to me at this particular location. John, I'm going to ask you a follow-up question. MRWC is doing a lot of advocacy kind of on the state level, but what can individuals do? How can they help advocate or promote, you know, reducing CSOs, or the importance of monitoring in the Merrimack?
Sure. Well, one of the things you can do is just check out our emails when we send them out. You know, we have a monthly list of advocacy issues that we're working on. And it really helps when people send in their own comments to the legislature. I mean, legislators read these things. And if they get 50 or 100 comments on a particular issue, it can make a big difference, and it can push a bill forward, like the bill I mentioned a few moments ago. So citizen advocacy is very strong, and it's great when we see people turn out like we just had for this CSO notification bill. That passed because people really in the Merrimack Valley demanded it, and so it happened. So keep those letters coming. Call your reps, call your senators. That's really effective. You know, read the news. You know, just type in Merrimack River news in Google every once in a while, see what's going on, and see if there's something you can be involved in. It's really all about staying in touch and then reacting, in a positive way when you see something. And we try to guide people to let them know, here's who to contact. And if you can follow up on that, that's a huge help to all of us.
Great. Yeah, I agree. Massachusetts, I would say, is moving much farther than New Hampshire has so far. So this also applies to those of us on the New Hampshire side of the river. Kind of in that vein, I'm going to send a question to Susie about CSOs in Manchester. So you had talked about how we don't have so much data from Manchester, but even that's changing and that's evolving. Can you talk about, what data was available for 21, what data could be available for 22, and how does the, what data we have impact our ability to analyze CSO events?
Sure. Yeah. And John, feel free to chime in if I miss anything or you have something to add. But we get data in different ways from the different wastewater treatment facilities. And the way that I've been collecting it for this report is through the annual reports that they put out and downloading those reports and the data that they make publicly available. So all of the wastewater treatment plants, except Manchester, make data available regarding each individual CSO event. So when that event occurred and how many gallons of combined sewer overflow went into the river, where it went in, they provide a lot of information. And so in our report, we had data for 2021 for all of the plants except Manchester and Haverhill. That's not because Haverhill doesn't put the information out, they just haven't put it out yet for this year or for this past year. Manchester, however, we don't have as much information for. So we have their annual total volumes, but we don't have information regarding each individual event, how big it was, exactly when the event happens. We don't get email notifications, or at least we haven't in the past gotten email notifications from them. However, we started to see some really great changes. So they have a new system now where we are receiving email notifications each time a CSO event occurs. So we're hopeful that means they're collecting the data to release regarding the volumes discharged in each event. So really excited to see that and hopeful that, by this time next year, we have more data to plug into our report than we had this year. John, did I miss anything? Are there more bits of information? John and I both kind of tackle the CSO data together.
Yeah,
so
right. Yeah, I know you hit it. That's great. Only one other minor thing to add, and that's that Manchester just adopted a system that Haverhill uses where you can see a live map of how much volume has come out of particular pipes. So that's helpful. Susie says we're not getting this day by day data, but you can look at that map and for a little while, you'll see how much volume came out. So that's a big advancement for Manchester. And I think that's one thing we see with all these plants is that they're ramping things up. They're almost a little bit out of sync with other plants. They're doing a better job, I think, in general, of trying to address what the public wants, but it costs money and they've got to sometimes change the way they do things. So we support them in trying to bridge these gaps.
Yeah, big thank you to our partners at those wastewater treatment facilities for all the work and the monitoring and the data sharing that they do. So that we're almost at the end. I'd like to kind of look forward maybe a little bit. So Chris, I know you work often with wastewater treatment plants. What are other parameters or other things that are coming up of concern, maybe based on new ways or new medications or new processing things? What are other things we should be looking at for water quality in rivers like the Merrimack?
Yeah, so I know the big hot topic issue are PFAS. That's the big hot topic issue right now. Currently, I know I can speak for the NWRA when I was working with the NWRA in the Boston area that they are coming up with a process to sample and monitor PFAS and wastewater effectively. Even on the environmental side up in New Jersey, when I deal with hazardous waste, PFAS is a new hot topic issue where you're seeing a lot of regulation coming down to regulate PFAS and the amount of it that is going into the environment and that businesses are using in general. So I know on the industrial side, which I can speak for, that regulatory bodies such as MWRA, your local municipalities, and also departments of environmental protection, they're kind of asking businesses to, look at what processes you're doing, look at what you're using in your processes as well to determine if they can be swapped out if they do contain PFAS as well. So PFAS is a hot topic issue that's, kind of ramping up more and more as the time progresses.
Yeah, great point. Jill, maybe do you have some insight on any PFAS issues in the Merrimack or activities that are happening around PFAS here?
Well, how much time do we have?
25 seconds.
Yeah, for folks who might not be familiar, PFAS or polyfluoroalkyl substances. It's a huge family of chemical compounds that help make things water resistant, grease resistant, and they're part of our lives. So they're in our soil, they're in our water, and they're in us. So there have been studies by the CDC showing that up to 97% of all humans have PFAS in their systems. That's nothing to be terrified about. We don't know a whole lot about dangerous levels, but we know that trace amounts are just part of our lives. So USGS and DEP did an interesting study just a couple of years ago of surface waters of rivers in our region, including the Merrimack, looking for PFAS. And they studied several dozen rivers and found them in all of them. So it's just a part of our reality. They are here and they're here to stay since they don't degrade in the environment. So the best thing that we can do is educate ourselves, be aware of safer products that you and your family can use. Look at your water testing results from your municipality or your city. Those are publicly available. I can look up Groveland's, and I just saw that last year we were at 10 parts per trillion, and the Massachusetts standard is 20 parts per trillion. So I'd really like it to be 0, but I'm comforted by the fact that we're below the threshold. So just inform yourselves, educate yourselves, but also don't lose sleep over it really at night.
Great. And with that, I think it's time to wrap up our webinar and our panel. I would like to give a big thank you to all of our panelists and Susie for your nice presentation. I think it's been a real nice discussion. I think we could go on for another hour talking about water quality. So if you all in the audience, if you have more questions, feel free to email us at volunteer@merrimack.org. And we'll get back to you and incorporate it into our future analysis and considerations as we move forward with our monitoring program in the future. So again, a big thank you to panelists, a big thank you to our participants for joining us this evening. Also our funders and our everyday donors. This couldn't happen without you. This takes a lot of time, effort, and money to make a program like this happen. So we want to make sure that we extend our gratitude to everyone for supporting this. And again, check out our website, check out our dashboards. We're very proud of them, and we hope it helps you understand the Merrimack a bit better and the water quality conditions. So hopefully we'll see you again in 2023 to talk about our 2022 results. So thanks again, and everyone have a nice evening.